Right. So welcome, everyone. Thanks for making time today. I'm very excited to introduce someone that I knew from graduate school, Rob Signer, who comes to us from Canada initially. He's a fellow hockey player. He studied engineering at the University of Toronto, and then he came to California for his Ph.D., working with Ken Dorshkin, where he made some exciting findings about blood cell development and aging and how it was relevant to disease. After working here, he went to postdoc with Sean Morrison, who at that time was at University of Michigan, a fellow Canadian. And then while he was in Michigan, the lab moved south to UT Southwestern in Dallas. And Rob made some incredible discoveries in Sean Morrison's lab, including a really fundamental finding about the regulation of protein synthesis in blood-forming stem and progenitor cells. He did so well that he got the opportunity to start his own group at UC San Diego, where he's been for the last 6 years or so. He's been just wildly successful in this time in San Diego, earning several R01s, a number of awards, V Scholar, other foundations, where he's an assistant professor in the Department of Medicine and the Division of Regenerative Medicine. In the last few years, his group has been able to show how critical it is to regulate protein synthesis for stem cell self-renewal and aging and disease, talking about the accumulation of misfolded proteins and how they can affect stem cell behavior, and recently addressing how this really critical problem of expanding stem cells ex vivo and how protein synthesis plays a key role in this, and this could be really important moving forward as we try to expand stem cells outside of the body for transplantation. So very excited to welcome Rob Signer to give a virtual talk, and we hope he can come back to campus soon. And so, yeah, please take it away, Rob.
Thanks so much, Andrew, for the kind invitation and introduction today. And, you know, of course, I wish I could be there in person to see plenty of old friends and colleagues, but, you know, this is the situation that we're still in, but I'm really excited to share some work with you today that will really review our studies on protein homeostasis in stem cells, and we'll also include a chunk of new unpublished data as well. So I'm going to go ahead and share my screen as soon as I'm able to do that. Great. So as Andrew alluded to in the introduction, the work in my lab really focuses on how hematopoietic stem and progenitor cells regulate proteostasis. So the first key question for a lot of people is, well, what is proteostasis? So as a word, proteostasis is a portmanteau of the words protein and homeostasis, and is really this idea that there's a network of biological pathways that regulate both the content and the quality of the proteome. So these pathways include physiological mechanisms that control things like protein biogenesis, protein folding, protein trafficking, and protein degradation. Now, an important thing that we've encountered while studying this, and I hope a take-home point for you today, is that this network is highly integrated. That is, if you perturb one node in this pathway, there can really be sort of complex compensatory changes in other nodes of the network that occur to rebalance proteostasis. Now, in some cases, this can be through direct changes, but in other cases, it can be through indirect changes that are driven by stress response pathways, such as the unfolded protein response in the ER or mitochondria, or through the cytoplasmic heat shock response. So why is proteostasis important? Well, the maintenance of proteostasis is really key for a number of processes. It's key to ensuring normal development, the resistance to environmental stress, and coping with infection, and probably most notably for regulating healthy aging and lifespan. So really an amazing observation has been that genetic or environmental interventions that enhance the capacity to maintain proteostasis will actually extend organismal lifespan in an evolutionarily conserved manner. That is, if you can improve an organism's ability to maintain proteostasis or to cope with protein stress, it will actually live a longer and healthier life. Now, in addition to this, the loss of protein homeostasis is really considered to be one of the hallmarks of aging. And the accumulation of misfolded proteins is associated with a number of degenerative diseases, most notably many neurodegenerative diseases. And for a long time, this is why proteostasis has really been focused on in the nervous system, and that people have thought that the reason that neurons are particularly susceptible to the accumulation of misfolded proteins is that they're long-lived post-mitotic cells. And so their long life allows them to accumulate misfolded proteins. And it turns out one of the best ways to eliminate misfolded proteins is by dilution that occurs through cell division. Well, hematopoietic stem cells are also very long-lived, and they're quiescent most of the time. So a central hypothesis that drives the work in my lab is that stem cells are also particularly dependent on the maintenance of proteostasis, and that disruptions in proteostasis contribute to stem cell dysfunction and aging. And overall, we really favor the idea that mechanisms that confer organismal longevity are conserved at the cellular level to maintain healthy, long-lived stem cells. So what I'll tell you about in my seminar today is really our efforts to unravel how the proteostasis network functions within hematopoietic stem cells. And I'll touch on at least 4 different components of this network today: translation, protein folding, protein degradation, and the heat shock response. And if there's one thing you take from my talk today, I really hope it's that many of these mechanisms, which are highly conserved essential physiological mechanisms, actually function and are coordinated differently in stem cells than in restricted progenitors, and are often coordinated in stem cells with the explicit purpose of promoting stem cell fitness and longevity. So I'm sure that many of you are already familiar with the hematopoietic system, but briefly for anyone that's not, the hematopoietic system is maintained by a rare population of hematopoietic stem cells that reside in the bone marrow. These rare stem cells have the capacity to self-renew throughout life, and they can differentiate in what's classically been described as a hierarchical fashion to give rise to all of the different types of blood and immune cells. Now, there are some great advantages, but also some disadvantages of working in this system. I'd say one of the key advantages is that we can identify and isolate each of these stem and progenitor cell populations with very high purity. Now, a couple of the downsides is, one, I mentioned that stem cells are very rare cells. And the other is that the biology of these stem cells seems to change quite dramatically when we take them outside of the body. And so when we want to interrogate stem cell function, we really need to assays that are tractable for use within small numbers of cells in vivo. And when we think about the protein homeostasis network, many of these exist for studying large numbers of cells in culture. And so what I hope you'll appreciate through the talk today is that there's been a huge amount of technology development that has had to go into our ability to study this within stem cells. And the first piece of technology that we adapted to do this was a molecule called O-propargyl-puromycin, or OP-Puro, that allowed us to measure rates of protein synthesis within single cells in vivo. And when we used this tool to look at protein synthesis, we made 2 sort of really interesting observations. The first is that contrary to what most people think of in regards to protein synthesis, that it's largely a housekeeping function that's the same in most cells, turns out not to be the case at all. That individual cell types all really establish their own specific homeostatic rate of protein synthesis. And that rate can go down or up even through the trajectory of differentiation in a single tissue. Now, the other really striking thing that we found was that hematopoietic stem cells, or their most closely related and primitive multipotent progenitors, have far lower rates of protein synthesis than in any restricted progenitor or differentiated cell population that we've looked at. And I'm showing you some of those here, and we've now probably looked at upwards of 15 different populations of cells in the blood. And this actually turns out to be a conserved feature of somatic stem cells across tissues. Now, the sort of simplest explanation for why stem cells have low protein synthesis would have to do with their quiescent nature. And so we wanted to test this directly. And so the way that we did this was by driving hematopoietic stem cells into cycle using a treatment of cyclophosphamide and G-CSF. So this is a treatment that is known to push stem cells into cycle. So under steady state conditions, you can see that very few hematopoietic stem cells are cycling. Roughly 10% are positive for the cell cycle marker Ki67. And after cyclophosphamide and G-CSF treatment, now nearly all of the stem cells are actively cycling. So we did this, we used this treatment and then measured protein synthesis rates using OP-Puro. And what we found was that when we looked within the non-dividing stem cells, the cells in G0 and G1, where we know the majority of these cells are actually now in G1 in the cell cycle, well, they still have lower protein synthesis than each of these restricted progenitor populations. And even if we look at the dividing fraction of stem cells, the cells in SG2 and M, well, the dividing stem cells still have lower protein synthesis rates than the dividing fraction of all of these restricted progenitor populations. And so this isn't just due to their quiescent nature, this is a true feature of hematopoietic stem cells. Now, it turns out this isn't just a feature of stem cells, it's also actually critical to their function. And the way that we tested that was by using a series of genetic mouse models. The first mouse model that we used had a hypomorphic mutation in the gene encoding the ribosomal protein RPL24. And you can see that RPL24 mutant stem cells have about a 40% reduction from that already low rate of protein synthesis in stem cells. The other genetic mouse model that we used was where we conditionally deleted the tumor suppressor PTEN. PTEN is a negative regulator of the PI3 kinase and mTOR signaling pathways that are known to promote translation. And you can see that the PTEN deficient stem cells have about a 30% increase in protein synthesis. And then what we did was we actually crossed these two mutant mouse strains together. And we found that the PTEN deficiency could partially rescue the decline in protein synthesis that we saw with the RPL24 mutation. But more notably, the RPL24 mutation was able to block the increase in protein synthesis that we saw with the PTEN mutation alone. So with these tools in hand, we tested stem cell function by performing a competitive stem cell transplantation assay. And what you can see here is the level of reconstitution you get in the black bar from wild type stem cells. When you reduce protein synthesis with the RPL24 mutation, you reduce stem cell reconstituting activity, and that's shown here. When you increase protein synthesis with the PTEN mutation, you completely lose long-term reconstituting activity. But remarkably, when you put these two mutations together, that each on their own impair stem cell function, they're able to mutually rescue each other. And what that's telling us is that stem cells have this low rate of protein synthesis, and modest changes in that protein synthesis, whether it's a modest decrease and especially a modest increase in protein synthesis, is really detrimental and in some cases catastrophic for stem cell function. So it was really at this point that we started, that I started my own group, and we really wanted to understand translation in the context of protein homeostasis. And the reason that we did that is because translation or protein synthesis is actually a highly error-prone process. So it is the most error-prone aspect of gene expression and is orders of magnitude more error-prone than, say, DNA replication or transcription. And in fact, it's been estimated that approximately 18% of proteins contain at least one amino acid that's been misincorporated. And between 10 to 20% of all newly synthesized proteins are immediately targeted for degradation. And so we wondered if the low rate of protein synthesis in stem cells was actually important for maintaining protein quality and homeostasis. And this work was really done by an awesome team in the lab led by former postdoc Lorena Hidalgo, as well as my terrific lab manager, Mary Jean Sunshine, a former technician, Chris Dillingham, and what's now a senior graduate student, Bernadette Chua, whose additional work I'll also talk about later in my seminar. So in order to look at protein quality in stem cells, we adopted what is most typically done within the protein homeostasis field. and that's to look at levels of polyubiquitinated protein by Western blot. So unlike a normal Western blot where you're really looking for a single band, what you're looking for in this case is sort of a smear of polyubiquitinated proteins. These are proteins that, for the most part, are being targeted for degradation and are basically a surrogate that's being used to quantify misfolded proteins in cells. And you can see clearly that stem cells have fewer polyubiquitinated or misfolded proteins, as compared to these restricted progenitor populations. But one of the flaws with this assay is it's impossible to quantify because you can't ever get this smear of polyubiquitinated protein within the linear range. And so what we did was we developed a method to detect this by intracellular flow cytometry where we could better quantify it. And when we did that, you could see that the stem cells have between 50 and 150% less misfolded protein than these progenitors. And this is true even if we normalize for total protein within these cells, which is actually relatively constant. Now, not all polyubiquitinated protein is misfolded, and so we really wanted an orthogonal assay to assess protein quality. And so we adapted a molecule called tetraphenylethyl malamide, which we just call TMI for short. So TMI is a cell-permeable dye that fluoresces when it binds to free cysteine thiols. So cysteines are highly hydrophobic residues that are typically found within the core of globular proteins, but can be exposed in unfolded proteins. And so when TMI interacts with these cysteine thiol groups, it fluoresces. And so we use this to measure unfolded protein within hematopoietic stem and progenitor cells. And similar to what we observed with polyubiquitinated protein, we saw that stem cells had less unfolded protein than each of these restricted progenitor populations. So at this point, we knew that stem cells had low protein synthesis, and we knew that they had few misfolded and unfolded proteins. But the question is, are these two things actually related to one another? And so to test that, we went back to our mouse models where we could alter protein synthesis rates. And again, you can see when we delete P10, we have an increase in protein synthesis. And that led to an accumulation of both polyubiquitinated and unfolded protein within these stem cells. We block that increase in protein synthesis with the RPL24 mutation, and we can block the accumulation of both polyubiquitinated and unfolded protein. And so that's telling us that stem cells really depend on low protein synthesis to prevent the biogenesis of these misfolded and unfolded proteins. Okay, so we know now that stem cells have low protein synthesis. This is protecting their sort of proteome integrity. But is it the accumulation of misfolded and unfolded proteins that's actually driving the stem cell defects when protein synthesis is increased? And so to test that, we actually needed a model where we could decouple protein synthesis rates from protein quality. And to do that, we turned to this mouse model that has a mutation in the editing domain of the alanine tRNA synthetase. And this mutation reduces tRNA editing and causes increased amino acid misincorporation, specifically with serines being incorporated in place of alanines. Now, these mice, you can see, have a little bit of a skin and hair texture defect, and they were termed the sticky mice because of this. And they've mostly been used because they develop an age-dependent neurodegenerative disorder, which is characteristic of proteostasis dysfunction. But no one had ever looked in a stem cell population or in the blood. So we obtained these mice and began to characterize the hematopoietic system. And you can see that the hematopoietic stem cells from these tRNA editing mutant mice do accumulate polyubiquitinated protein. But importantly, we actually don't see much of that accumulation within these progenitors. And we think that this reflects the proliferative nature of these progenitors, that they can dilute out these misfolded proteins. Another important point here is that the accumulation of polyubiquitinated protein in these stem cells is really within the physiological range. We're actually just making stem cells look more like progenitors in terms of their amount of unfolded or misfolded protein. And importantly, we're able to do this without altering overall rates of protein synthesis, which we show to be basically the same between these wild type and these tRNA editing mutant mice. So now we had a model to assess the impact of protein misfolding on hematopoietic stem cells, and we characterized the hematopoietic system in these mice. we found that these mice had about a 40% reduction in both the frequency and absolute number of hematopoietic stem cells, but restricted progenitors were mostly unperturbed or unchanged. And so we tested now stem cell function again by using a competitive transplantation assay. And you can see actually, and somewhat to our surprise, that these stem cells function quite normally as compared to their wild type counterparts. And so what we then did at the end of this transplant is we performed a secondary transplant, which is really the key assay for assessing stem cell self-renewal activity in vivo. And when we did this, we found a severe defect in reconstitution in these secondary recipients, indicating to us that this accumulation of misfolded protein or disruption in protein homeostasis was severely impairing stem cell self-renewal. From here, we wanted to understand more about the mechanism of how unfolded or misfolded proteins were driving stem cell dysfunction. And so we thought about this in two directions. We thought, well, what happens to these unfolded or misfolded proteins? Well, one thing is that they can trigger activation of these stress response pathways. But we actually found no evidence of this in the context of these tRNA editing mutant mice. And so we considered the other direction that these things can go, in that there are mechanisms to degrade these unfolded and misfolded proteins in stem cells, and maybe this was impacting protein degradation. And so the canonical pathway, or at least what's thought to be the canonical pathway for degradation of misfolded proteins, is the ubiquitin proteasome system. And so we measured proteasome activity in stem and progenitor cells. And similar to what we saw with protein synthesis, we saw that stem cells had much less proteasome activity as compared to these restricted progenitor populations. And you can note here that this is on a log scale. So these are quite large differences between these cell populations. And so we thought, well, if stem cells have low proteasome abundance, is it possible that these misfolded proteins are actually overwhelming the capacity of stem cells to degrade these products? And again, to test that, we needed another mouse model. And we used what are called these G76v Gfp mice. Now, these mice ubiquitously express an unstable form of GFP. So basically, every cell type in the body will transcribe and translate GFP, but because it's unstable, it's rapidly degraded. So you can see the GFP transcript, but you can never really detect the fluorescence because of the rapid degradation. And the reason that these mice even exist is to screen for proteasome inhibitors. So if you administer a proteasome inhibitor to these mice, or put the cells, grow them in the presence of a proteasome inhibitor, like is shown here, all of a sudden you can see GFP light up in these cells. So we crossed these mice to these tRNA editing mutant mice, and lo and behold, we could see GFP begin to accumulate in some of these stem cells, really suggesting to us that the accumulation of misfolded proteins was overwhelming the degradation capacity in stem cells. Now, what's the consequence of overwhelming the proteasome? Well, it's known that there are actually many key regulators of stem cell function that are regulated post-translationally by the proteasome. And one of those key factors is c-Myc. So work from Ioannis Ifontis's lab, as well as Andreas Trump's lab, has really characterized a role for Myc in hematopoietic stem cells. So Myc is transcribed and translated within the stem cells, and then is really controlled at the level of degradation. And levels of Myc protein can really dictate fate determination of these stem cells. So if you have an abundance of c-Myc protein, and they showed this by either overexpressing it or deleting the ubiquitin ligase that targets it for degradation, it will drive stem cells into cycle. And ultimately, if that's sustained, will promote their differentiation. But if you delete Myc entirely, those stem cells have a defect in differentiation and can't proliferate properly. So you can see when we looked at levels of Myc protein within these tRNA editing mutant stem cells, where the proteasome is overwhelmed, we could see Myc protein accumulate. Again, this was specific to stem cells. We never saw this within progenitors that normally have much higher levels of Myc protein, and that this isn't due to a transcriptional change in Myc, which was unchanged when we looked at it either by RNA sequencing or by qPCR. And consistent with the known role of Myc in hematopoietic stem cells, we saw increased proliferative activity of these tRNA editing mutant stem cells. And so next we actually tested what happened, to what extent Mick was contributing to these phenotypes. And so we crossed this to a mouse where we conditionally deleted a single allele of Mick from the hematopoietic system. And the reason we did a single allele is because complete loss of Mick will impair stem cell function. I think we got a little bit lucky here in that deletion of one allele of Mick was able to bring Mick protein levels back down to near wild type levels. And then again, we performed the serial transplantation assay. And indeed, we found that by reducing the abundance of Mick protein, we could largely rescue the serial reconstituting and self-renewal defect that we saw within these tRNA editing mutant stem cells. Now, all of this work at this time actually led us to ask another question, and that is why we weren't actually seeing stem cells activate any of these stress response pathways when they accumulated unfolded or misfolded proteins. And we really wanted to know what are the mechanisms that regulate stem cell proteostasis in response to stress. And our work here was really led by a former postdoc, Mariama Kruta. And our inroads into this actually came from an experiment that we were doing to try to understand why it's so difficult to grow hematopoietic stem cells ex vivo in culture. And the experiment that we did in this regard was to try to understand what the intrinsic changes are within stem cells in culture. And so what we did was we isolated stem cells and grew them in culture under sort of the best conditions we had for their maintenance for a period of 18 hours, and did RNA sequencing comparing that to just freshly isolated stem cells. And I joke sometimes that this was probably a terrible experiment because it turns out that there are just massive changes in gene expression that occur within these first 18 hours in culture and stem cells, with over 2,000 genes showing a significant twofold or greater change in their expression. But maybe we got a little bit lucky here as well, and that's that some of the most upregulated pathways in culture really had to do with protein synthesis. So we saw massive upregulation of things related to ribosome biogenesis, mRNA processing, translation, initiation, elongation, termination. And we also saw a big upregulation of this response to heat gene signature. And so together, this raised the possibility to us that stem cells in culture were experiencing large increases in protein synthesis, and that this might actually be activating the heat shock response. And so we went on to test this directly. And so we measured protein synthesis within hematopoietic stem cells in culture, and the results are really striking. So within just 4 hours in culture, we see nearly a 700% increase in the rate of protein synthesis. And after 18 hours, we see nearly a 2,000% increase in protein synthesis. And this is not just due to the fact that we're activating stem cells in culture, because if we go back to our cyclophosphamide and GCSF experiments, where we know we're pushing stem cells to undergo rapid division in vivo, we do see a small increase in protein synthesis, but it's still an order of magnitude different than what we see within our cell culture system. And as a reminder, a sustained 30% increase in protein synthesis is catastrophic for stem cell function in vivo. So we certainly thought a 2,000% increase was really just this massive stress. Now, in accordance with what we saw in vivo, the increase in protein synthesis within cultured stem cells was also leading to this accumulation of misfolded proteins. So already within 4 hours, you can start to see that accumulation here. By 18 hours, you just have this overwhelming accumulation of polyubiquitinated or misfolded protein in cultured stem cells. And this is definitely due to de novo protein synthesis, because if we add in a translational inhibitor like cyclohexamide, we can completely block that accumulation of polyubiquitinated protein. So protein synthesis is massively increased. There's this massive protein stress in culture. Are we now really activating a stress response pathway? And so the heat shock response is the major cytoplasmic pathway that responds to proteostasis dysfunction and is really regulated by a master regulator transcription factor called heat shock factor 1 or HSF1. Now, under steady state conditions, HSF1 is typically sequestered in the cytoplasm through its interaction with chaperones like HSP90 or TRIC. Now, under conditions of protein stress, what happens is that the chaperones will dissociate from HSF1, bind to these unfolded or misfolded proteins. This allows HSF1 to translocate to the nucleus, where it will function as a transcription factor to drive transcription of things like heat shock proteins or other things that can help to restore balance to protein homeostasis. Now, when we looked at HSF1 activation by looking at its nuclear accumulation, you can see that in young adult stem cells, we rarely see HSF1 significantly accumulate in the nucleus. This is consistent with our observations that these stem cells have low protein synthesis and very few misfolded or unfolded proteins. In contrast, though, when we put the stem cells into culture, now after 18 hours, we can see HSF1 accumulate within the nucleus of these cultured stem cells, and the quantification of that is shown down below. And so this raises a question of, well, what is HSF1 doing under these conditions in stem cells? Is it protective for stem cells? Is it contributing to our inability to grow stem cells? And so to test this, what we did was we conditionally deleted HSF1 within the hematopoietic system. And then we isolated stem cells from either these HSF1 deficient mice or wild type controls. We seeded them in 96 well plates at a density of just 10 stem cells per well. We grew them in the best conditions that we had at the time and left them unperturbed for a period of 10 days. At the end of 10 days, we harvested the entire contents of that well, so whatever those 10 stem cells gave rise to, and we competitively transplanted them with 200,000 freshly isolated bone marrow cells into irradiated mice. Now, first, what you can see here is that HSF1 deficiency does not impair the ability of these stem cells to proliferate in culture. So these 10 cells after 10 days will give rise to between 20 and 30,000 cells. That's, at least some heterogeneous mixture of potentially stem and progenitor cells. But when we do the transplantation experiment, you can see that HSF1 deficiency severely impairs the long-term reconstituting activity of these cultured hematopoietic stem cells, telling us that HSF1 is functioning to promote stem cell maintenance in culture. Importantly, this does not reflect a requirement for HSF1 by young adult stem cells in vivo. So if we just freshly isolate stem cells from these HSF1 knockout animals, they reconstitute similarly to wild type stem cells, and that's true in both primary and secondary transplants. And so at this point, we wondered, well, if HSF1 is promoting stem cell maintenance in culture, is there a way for us to enhance or super activate this pathway, and will it improve our ability to grow stem cells? And so small molecule inhibitors of either HSP90 or TRIC had been previously shown to be able to block the interaction with HSF1 and enhance HSF1 activity in certain cell types. We showed that this activity was operative within stem cells. So you can see the cultured stem cells here have a little bit of HSF1 in their nucleus. When we grow them in the presence of either an HSP90 or a TRIC inhibitor, we can significantly increase that HSF1 that's localized to the nucleus. And so now we could do the experiment where we could test if growing stem cells in the presence of either an HSP90 or a TRIC inhibitor could actually improve stem cell maintenance. And so we did this again by seeding 10 stem cells for 10 days in the presence of either of these inhibitors or our vehicle control, performing that competitive transplant. And in this case, we also performed a secondary transplant. And so what you can see here from our primary transplant is that the cells grown in the presence of this HSP90 or TRIC inhibitor definitely show a trend toward having higher reconstituting activity as compared to these controls. But sort of remarkably, the major difference showed up in secondary transplants, where now these cells that had initially been cultured in the presence of either these HSP90 or TRIC inhibitors showed 3 or 4-fold times higher levels of reconstituting activity in secondary transplants. And when you look overall at the number of long-term multilineage reconstituted mice, you can see that under our normal culture conditions, we can reconstitute about 1/3 of these recipients, we can boost that to 2/3 with the TRK inhibitor and up to near 85% with the HSP90 inhibitor. And so this was telling us that we were really improving our ability to maintain stem cells. But there were sort of two questions here. Was this because we were improving or enabling expansion of these stem cells in culture, or were we actually just improving the quality of these stem cells? And so we did limiting dilution transplants where we found it turns out we actually weren't increasing the number of stem cells. Those 10 stem cells stayed at about 10 stem cells, even under these conditions where they were giving rise to 20 or 30,000 progenitors. And we wondered, well, then how good are these stem cells really? And so we compared our cultured stem cells to freshly isolated stem cells. And quite remarkably, we found that our cultured stem cells were actually giving higher levels of reconstitution, even than equal numbers of freshly isolated stem cells. And that was sustained to have equal activity even through secondary transplant, telling us that this activation of HSF1 was really promoting the maintenance of stem cell fitness in culture. Now, I won't show you all the data that we've done because it's published, but based on the role of HSF1 and some RNA-seq data, we wanted to investigate the role of HSF1 in promoting proteostasis maintenance in cultured stem cells. A little bit surprisingly, because it doesn't have a known role in translational control, we found that HSF1 deficient stem cells had even higher protein synthesis rates than just our wild-type cultured stem cells, and that we could significantly reduce protein synthesis rates with the HSP90 inhibitor. We also assessed proteostasis again using this G76v Gfp mouse that I introduced earlier. So in culture, you'll see GFP accumulate in these mice, demonstrating proteostasis imbalance. This is exacerbated by HSF1 deficiency and significantly improved when we sort of superactivate HSF1 in the presence of the HSP90 inhibitor. Now, we were really excited about all this work in culture, but we still were wondering, well, is there actually a physiological role for HSF1 in vivo? And given sort of the known connection between proteostasis and HSF1 and aging, we looked in that context. And what we found was that in contrast to young adult stem cells, by middle age, we could already see HSF1 beginning to accumulate within the nucleus of these of stem cells in 1-year-old mice. And similar to what we saw in culture, we found that HSF1 was actually protecting proteostasis by restricting protein synthesis. So you can see in young adult mice, there's no effective HSF1 loss, but by 1 year of age, HSF1 loss significantly increases protein synthesis within stem cells. When we measure stem cell activity in transplantation assays, you can see that a loss of HSF1, even though it wasn't required for young adult stem cells, HSF1 now is actually promoting stem cell activity in middle-aged mice. And since we've published this work, we continue to be interested in the role of HSF1 in aging. We've now actually have data from 2-year-old mice where this seems to be further accentuated, where a loss of HSF1 in old adult mice is really detrimental, where we only see long-term multilineage reconstitution about 15% of the time. And so this is telling us that there is protein stress that is accumulating within aging stem cells, that they're activating HSF1. HSF1 is helping to maintain proteostasis balance during aging and is promoting the maintenance of stem cell fitness during aging. Now, the last thing I want to tell you about today is some unpublished work, and I mentioned this earlier, from now a senior graduate student and former UCLA Bruin, Bernadette Chua, and she's just done a heroic work on this project, generating just a ton of data and overcoming all sorts of roadblocks. So it's been really awesome as her PI to see this unfold over the course of a number of years. And what this project was to really understand the role of protein degradation in regulating proteostasis. Because there was one thing that was really bothering us about our data, and that is that if proteostasis maintenance is so important for stem cells, why do they have such low proteasome activity? Wouldn't we expect them to have high proteasome activity that would allow them to sort of buffer against any accumulation of misfolded proteins? And so the idea that we had here is, well, even though the proteasome is sort of this canonical pathway for eliminating misfolded proteins, maybe stem cells are using an alternative pathway. Maybe they're using autophagy. And so the major form of autophagy that I'm talking about is called macroautophagy, where material is engulfed within double membrane vesicles called autophagosomes, which can fuse to lysosomes to form autolysosomes. So there's been really outstanding work from Katja Simon or Emmanuel Pasquier and others that's demonstrated an important role for macroautophagy within hematopoietic stem cells, with particular demonstration that stem cells robustly activate autophagy in response to stress, and that degradation of mitochondria or mitophagy is important for stem cells. But we still know very little about steady state autophagic activity in stem cells, and to what extent it influences proteostasis is completely untested. And so we set out to quantify autophagy within hematopoietic stem cells in vivo using reporter mice in which LC3 is fused to both GFP and RFP. So LC3 is a cytosolic protein that gets modified and localizes to the surface of autophagosomes. And the dual fluorescence reporter here is extremely useful for examining autophagy because it not only reports on LC3 expression, but actually reports on its flux through the autophagy pathway. And that's because GFP gets quenched in the acidic lysosomes, but RFP does not. And so we can actually examine cells from these mice by flow cytometry and see three categories of cells. We can see RFP, GFP double negative cells that have no ongoing autophagy. We can see RFP, GFP double positive cells that contain autophagosomes, and we consider these cells to have low autophagic flux. And we can see RFP positive, GFP negative cells that have autolysosomes present, and we think thus exhibit active or high autophagic flux. And so when we did this within the hematopoietic system, we found that restricted progenitors really have very little ongoing autophagy, but that roughly 75% of stem cells have active ongoing autophagy, with the majority of these actually having very high autophagic activity at steady state. Now, I think the best way to actually view this is with these histograms, as they make two really important points. One is that there are more HSCs undergoing autophagy as compared to progenitors. And two, on a per cell basis, HSCs are actually undergoing more autophagy, so the fluorescence level is higher than even these rare autophagic progenitors. So, to us, this was a very exciting finding and raised the possibility that stem cells might have high autophagic activity and that they preferentially use this to help maintain proteostasis. Now, before I show you data on proteostasis, we wanted to understand a little bit more about the differences between these autophagic and non-autophagic stem cells. And so what we did here was we actually sorted and transplanted stem cells from each of these different sort of autophagic subsets, the high flux, the low flux, and the no flux. And what we found was that they gave sort of similar levels of long-term reconstitution. So functionally, these all appear to be fairly equivalent. They all give rise to stem cells in the bone marrow of these transplant recipients. But we did learn something interesting from these experiments. And that is that regardless of which subset of stem cells we transplanted, whether they were the high flux stem cells, the low flux stem cells, or the no flux stem cells, they all gave rise to each of these subsets once again in these recipients. And so that's telling us that there's plasticity between each of these states, and that there isn't just a linear order of differentiation that occurs between them, and that these cells are fluxing between having low, high, or no autophagy at any given time. The second thing that we learned is that after transplantation, there was a pretty striking change in the distribution of these stem cells, where now these no flux or these high and low flux stem cells were the minority, but these no flux stem cells took over as the majority. And we continue to try to understand, how this transplantation stress has influenced this change in autophagy. Now, even though there were no obvious functional differences between these subsets, we did identify molecular differences when we did RNA sequencing on each of these subsets. We actually found that the low and high autophagic flux stem cells were very similar transcriptionally. There were very few differentially expressed genes, But when we compared these high or low flux stem cells to the no flux stem cells, we found between 200 and 300 differentially expressed genes. And so there was something actually different about these stem cells, in that the pathways that are most really upregulated in these no flux stem cells are really ones related to having increased metabolic activity. So you can see they have increased gene signatures related to oxidative phosphorylation, as well as a lot of biosynthetic pathways, including ones that we're interested in ribosome biogenesis and translation. And to go along with this, we actually find that these no flux stem cells are basically twice as likely to be in the cell cycle as compared to the autophagic stem cells. So this does represent some difference in terms of the stem cell activation status here. Now, the other thing we wanted to confirm was that autophagy was really important for stem cells. And the way that we did that was by conditionally deleting the essential autophagy gene, ATG5. When we do that, you can see that we severely lose long-term reconstituting activity when we perform these competitive stem cell transplantations. So at this point, we know that stem cells have high autophagic activity. There's some changes in flux that are ongoing between these cells. And we know that autophagy is critically important for stem cells, but to what extent is it regulating protein homeostasis? And so we tested this by genetically disabling autophagy, again, through conditional deletion of ATG5, and also looked at this in the context of proteasome inhibition by administering the proteasome inhibitor bortezomib. And a little bit to our surprise, we found that disabling autophagy or the proteasome on its own did not result in any significant accumulation of either unfolded or misfolded protein. But when we did these two things together, when we administered bortezomib to an ATG5 deficient mouse, well, now we find significant accumulation of both unfolded and misfolded protein. And what was actually really nice confirmation of this result is we could see this cooperation transcriptionally by RNA-seq. So neither ATG5 deficiency or proteasome inhibition on its own is sufficient to induce activation of the unfolded protein response, but combined disruption of these pathways did do that. And so this result raised a key question. Do both of these pathways really just similarly or redundantly contribute to proteostasis maintenance in stem cells, or are there distinct roles of these pathways? And is there a preference for one of these pathways over the other? And so to understand sort of potential crosstalk between the proteasome and autophagy, we looked at what happens to one when we inhibit the other. So you can see when we administer a proteasome inhibitor, we can actually further increase the level of autophagy in stem cells. And in fact, we see this trend across all progenitors, so that really autophagy serves as a sort of backup or stress response mechanism when the proteasome is inhibited. Now, on the flip side, when we disable autophagy, we actually see a significant increase of proteasome activity within stem cells, but we don't see this within restricted progenitors. And this makes sense when you take into consideration that these progenitors are minimally utilizing autophagy at steady state. And so disabling the pathway really shouldn't induce or require any compensatory response. Okay, so there is crosstalk between these two pathways, but how do we know if one pathway is sort of the primary pathway or the preferred pathway? And so the answer came to us by doing RNA-seq on these different subsets of either ATG5 or bortezomib treated mice. And when we did combined inhibition, we actually blocked both pathways. One of the genes that we saw to be most significantly increased is this small heat shock protein, HSPB8. Now, HSPB8 interacts with another protein as part of what's called the BAG3 complex. So BAG3 induces assembly of a complex consisting of HSP70, HSPB8, and CHIP. And BAG3 will actually mediate client ubiquitination and delivery of misfolded proteins into agrosomes. And agrosomes are these inclusion bodies that are found within cells and are known to be preferentially degraded by autophagy. And the abundance of BAG3 is really important for this process because it competes with another co-chaperone called BAG1, where when BAG1 is part of this complex, it will preferentially drive delivery of this cargo to the proteasome. And so when we looked in the situation where we were inhibiting autophagy in the proteasome, as I already showed you, we saw significant upregulation of HSPB8. Similarly, we saw significant upregulation of BAG3, but no change in BAG1. And this ratio of BAG3 to BAG1 is elevated under these conditions, suggesting to us that the stem cells might actually preferentially send their misfolded proteins toward agrosomes. Now, when we go back and look in steady state, mining some of our old RNA-seq data, we can actually see indeed that stem cells are actually the only population that we've looked at that significantly expresses HSPB8 at steady state, and that the ratio of BAG3 to BAG1 is much higher in stem cells than in any of these restricted progenitor populations that we've looked at. Again, suggesting that stem cells are preferentially shuttling these misfolded proteins towards agrosomes. In agreement of this, when we look at the abundance of protein aggregates or agrosomes in stem cells as compared to progenitors in the context of ATG5 deficiency, where autophagy is lost, you can see that when we lose autophagy, we have a significant accumulation of these aggregates within stem cells, but no effect within these restricted progenitors. And this, again, is consistent with the lack of their autophagic activity, and here consistent with stem cells depending on autophagy for the purpose of removing these protein aggregates. And degradation of these agrosomes is strictly dependent on autophagy in stem cells, as proteasome inhibition does not affect the accumulation of either aggregates or agrosomes. So taking all of these data together, it really appears that stem cells preferentially use autophagy for the purpose of what we think is controlling resource allocation. So when misfolded proteins are degraded by the proteasome, this occurs one protein at a time, with raw materials and amino acids immediately coming out the other end. However, if you preferentially store these misfolded proteins as aggregates, you can all of a sudden get a burst of resources when you need them by activating autophagy in lysosomes, which really makes sense for stem cells that are largely quiescent, but need to be responsive to bursts of proliferative stimuli or in response to injury or stress. Now, I know we're running late on time, so I want to show you one final piece of this puzzle, and that is, well, if autophagy is really preferential for eliminating misfolded proteins here, what exactly is the proteasome doing in stem cells? And we thought, well, it's not just misfolded proteins that need to get degraded. There is normal protein turnover that must occur. Now, measuring protein turnover in stem cells in vivo is exceedingly difficult, if not impossible right now. And so we turned to cell lines in collaboration with Eric Bennett's lab to do this. And we did this using this RPE1 cell line where we altered growth rates by altering serum concentrations, where we could grow cells that had now low protein synthesis, sort of a medium level of protein synthesis, or high protein synthesis, and measuring protein turnover using a Silac switch assay. Now, when we did this and quantified decay rates or protein turnover rates, we found something pretty interesting. And that is that even though we can vastly change these growth rates, the decay rates stay relatively constant. And that's telling us two things. So that this decay rate or protein turnover and growth rate are much more closely matched in slow-growing cells with low protein synthesis. And that this raises the possibility that protein degradation plays a greater role in limiting biomass and cell growth in slowly dividing cells with low protein synthesis, like stem cells. So I know this is confusing, so bear with me through one more slide, and I think I can explain this a little bit more clearly. So there is an intrinsic relationship between cell growth and division. That is that cells need to grow to divide, and that is that they increase typically biosynthesis so that they duplicate their proteome so that daughter cells are sort of similar in size to their mother cell. Now, if you think about the situation with quiescent stem cells, they have low protein synthesis, they have low protein degradation. These things are relatively in balance. When this is in balance, the cells don't grow. Think of this as having a bathtub that the faucet is dripping a little bit, but the drain is open. The bathtub is never going to fill up. Now, normally the way that we grow is that we increase biosynthesis. So think of this as turning up the faucet in your bathtub. even if the drain is open now, the input is greater than the output and the bathtub will begin to fill up. But there's actually another way to fill up the bathtub, right? And that is by eliminating protein degradation. So even if you have just this slow drip, if you block this outflow, now all of a sudden the bathtub will fill up again. And so this raised the question to us of whether proteasome might actually be limiting the ability of hematopoietic stem cells to grow. And so we tested this by administering the proteasome inhibitor to mice and assessing cell size. And you can see that stem cells are significantly growing when we do this, but not restricted progenitors. We can actually measure total protein content in this case, and you can see that the amount of protein is actually increasing within these stem cells, and that this is actually driving the stem cells to divide. So proteasome inhibition will actually drive stem cells into cycle. And that's telling us that under normal conditions, the proteasome is there and matched to protein synthesis to really help limit cell growth and to enforce stem cell quiescence. Importantly, that when we drive stem cells to divide with a proteasome inhibitor, it doesn't just fate them for differentiation. So when we do the transplantation after proteasome inhibition, we see that this acute inhibition still allows these dividing stem cells to reconstitute mice comparably to untreated mice. And so now we think we have a more complete picture of how these two protein degradation mechanisms are functioning in stem cells, with autophagy primarily there to control for misfolded and aggregated proteins and to control resource allocation to support growth, whereas the proteasome is there to just be involved in normal protein turnover and to help to enforce quiescence within stem cells. So I know I'm at time here. Let me quickly, again, thank everyone in my group who's contributed to this work. I think I called them out individually as we went along, but we really just have this tremendous group of people. As I was saying earlier today, the best part of my job is really working with this incredible team. Let me also thank our various collaborators and of course our sources of funding for support. And if for people that are willing to stick around a few extra minutes, I'm definitely happy to take questions. Thank you.
Thank you, Rob. That was outstanding. I love how you explain things so we can actually follow along, and it's clear you've always been extremely thoughtful and actually have very original ideas here. So I assume everyone who stuck through that was kind of racing to think how this applies to our own systems. I'm curious to ask the first question. When you take cells ex vivo and you were seeing very rapidly an increase in protein translation, does that suggest that in vivo there's some restrictive mechanism that's missing? And what do you think that is?
Yeah, that's a terrific question. And we do think that's the case, that there is something restrictive, because, you know, we've done this and others have done this, at least to our best ability in stem cells, to try to measure ribosomes. And, you know, we've had to do that by measuring, you know, specific ribosomal components. And it doesn't seem that ribosomes are sort of scarce in stem cells. They seem to have, you know, abundance of ribosomal RNA, which is typically the limiting factor for ribosomes. So we don't think that it's there. You know, we published one paper showing that differences in mTOR signaling and specifically regulation of EIF4E binding proteins was contributing to part of this restriction. Didn't have time to talk about that today, but that's only a piece of it. We think that there are several other mechanisms at play here, and we're chasing down some of them, although it's too early to say for me, you know, if we're right or not yet. So stay tuned there. But we do think there is, like you said, a restrictive mechanism happening to restrain ribosomal activity in stem cells.
And then with regards to that tRNA editing mutant, which had increased c-Myc and increased proliferation, do you know what's causing the Myc expression and the proliferation?
Well, so Myc is normally transcribed and translated in stem cells, and then in large part because of FBXW7 and ubiquitin ligase, it's targeted for degradation. And so basically it's there, it's being targeted for degradation normally. And what we think is that, you know, you have too many misfolded proteins, they're either overwhelming the proteasome or potentially inhibiting the proteasome. And now you can't degrade MYC as efficiently as you would normally. And so MYC protein is just allowed to build up in those stem cells. And that's going to drive stem cells into cycle.
Got it. There's a question about common lymphoid progenitors. So I know you showed a lot of different subsets, but have you found anything interesting in lymphoid progenitor cells?
So, I know somewhere out there, Ken is shaking his head that I haven't been looking more in lymphoid progenitors. We did once look at common lymphoid progenitors, and they also, they do have much higher protein synthesis than hematopoietic stem cells, although we haven't looked at all of these. We tend to focus more on myeloid progenitors in this context because they're rapidly dividing. We have really stark differences in protein synthesis that allows for this comparison. And also, one of the challenges that we've gotten into when we've tried to look at lymphoid cells is that as those lymphoid cells differentiate, they become much smaller. And so you're dealing with even less protein content, and there's normalization issues there. So we've focused a little bit more on myeloid progenitors, but we probably should be also looking at lymphoid progenitors. And I know now Ken is shaking his head, positively about that comment.
So here's a question about the PTEN knockout that impairs the HSC function. And the question is, do you think it does it by blocking autophagy?
Yeah, so it's something we've thought about recently going back to this. So we never looked at autophagy in that context, but we do know that that translation is a key driver of that defect there, because when we specifically block that increase in translation with the RPL24 mutation, we're able to rescue the function. And importantly, I didn't show it in this talk, but it's in the paper. The RPL24 mutation is not blocking the overall sort of hyperactivation of the PI3 kinase signaling pathway. So presumably everything that's happening downstream of PI3 kinase pathway activation is still happening. But that would, of course, that would include, potentially inhibiting autophagy, although we haven't tested that directly in that context.
Great. And then we'll take one last question. Are there cues from the microenvironment that contribute to proteostasis of HSCs in vivo?
So none that we've looked at specifically. There was some work from Lev Silberstein and David Skadden that identified one of those. We're starting to get more interested into what is not only potentially in the environment that might be driving effects on proteostasis, but how changes in proteostasis in stem cells might conversely impact the environment. So that's sort of an area that's on our radar that we want to address in sort of future studies, but haven't comprehensively looked at. We've been focused on intrinsic.
Excellent. All right, well, we'll call it there. Thank you so much, Rob, for an outstanding talk and great questions. And we look forward to having you on campus sometime soon.
Great, thanks for having me, and thanks again to everyone for the attention and questions.
Bye, everyone.